Hello, good afternoon, good morning, and good evening, depending on where you are. My name is Luciano Lucas. I'm the product manager for Bitplane, and today I'm introducing a very exciting series of webinars that start today, and we'll have them running for the next 3 to 5 months, depending on how many topics we will cover in the end. But for now, we have 3 topics planned to be covered, and the first one is today by Gabriel Martins. Is going to talk about his project, Haeckaliens and OPenT. That comes in a moment. The second one will be focusing on light sheet microscopy, and that will be in July. And the third one will be later in September, and it will focus on tissue clearing. So stay tuned for those exciting webinars. We have a hashtag, which is when size matters. And you can follow all the updates on this on our website, which is bitplane.com/sumo, as in sumo wrestler. All right, so without further ado, I'd like to introduce the 2 speakers today. We have Dieter Gohlmann from Bitplane. He has studied biology at the Albert Ludwig University in Freiburg in Germany, and he finished his studies in 1995. And at that point, he did a thesis in neurogenetics, axonal pathfinding in the optical lobe of Drosophila. And soon after that, he joined Bitplane, firstly as a salesperson, but very quickly transferred into the technical and application support team, where he has been for over 10 years. He is one of our most gifted and most expert Imaris users and experts within Bitplane, and we have him today illustrating what can be done with the types of images that Gabi can create with his imaging system. So that is Dieter Gohlmann. And the keynote speaker today is Dr. Gabriel Martins. He was born in Bragança, that's in Portugal, in the north of Portugal, in 1971. In 1994, he graduated from the University of Lisbon, the Faculty of Sciences, with a degree in biology, and he followed that with a post-graduation in developmental biology at the University of Lusófona, and also in the US, so it's part-time in one place, part-time in the other place, sunny Buffalo. Later, he studied the mechanics of somatogenesis at the University of Lisbon, and he has been invited recently to serve as the imaging specialist at the Institute Gulbenkian of Science, the cell imaging unit, which is based in Oeiras, just outside of Lisbon. He has authored several articles on developmental and cell biology and on microscopy imaging. One of his latest pieces of work is part of the OpenSpin project. That's OpenSpin with an N at the end. And this is a very exciting project that brings together 2 very, very exciting technologies. One is light sheet microscopy, and the second one is OPenT, which is what Gabi is going to be talking about today. So without further ado, I'd like to introduce or welcome Gabi to the floor. And I wish that you all enjoy this great presentation. By the way, Gabi did win the Nikon Small World in Motion Award for 2013, which was just announced about a month ago. And it was exactly with the, I think it was exactly with the image that he has on his slide. So many congratulations, Gabi, and I look forward to hearing your talk today.
Thanks a lot, Luciano, and everyone. First of all, I would like to thank you and the Bitplane team for offering me this opportunity and hosting this meeting. I also like to welcome everyone and thank you for your time. I hope I can convince you that this is really an interesting technology and that it's easy to get into. So the title of the talk is kind of strange. I'm aware that many of you may have actually started using optical projection tomography for a long time ago, even before probably I realized what it is. I came across it a few years ago and immediately realized that it was a technique that I'd like to have. The problem is you're probably aware that the economical situation here in Portugal has been complicated, and it was tough to get financing for research. So what I actually did was encouraged by my colleagues and actually by the inventor of optical projection tomography, I decided to build my own system. But before we move on to that, maybe I should tell you just a little bit more about why I did this and what I do. As Luciano said, I have worked on cell biology and morphogenesis. I'm an embryologist, and I was always very interested in how tissues form. And most developmental biologists, until recently, have focused on looking at cell-cell interactions and how those eventually lead to forming tissues and all organs. And we've known quite a lot about internal anatomy, or at least mostly about external anatomy of embryos. It's quite fascinating to see how they develop over time, how they change in shape, and how they come to be the organisms that we later recognize as adults. But most developmental biologists, for limitations of the optical microscopy itself, have not had access to images or 3D images and images of a lot of detail of the later stages of embryos. And I really got interested into it a few years ago, and the reason was, and I'll explain a little bit more later, is that I was really interested in understanding differences between species, different species, and how they vary during the development, and also within the same species, how certain characteristics and organs are formed, and how the rate at which they develop. For example, in one of the projects that I have been trying to get funding to, we're trying to understand how two embryos, for example, of a quail or of an ostrich, which start very similar in early development, leads to the appearance of an organism that is very different in size. And in fact, the title of the series of meetings that Luciano and Bitplane created fits just perfectly with what I'm interested in. Size matters. It's actually the title of the project that I've been trying to get approved. So let me start by telling you a little bit how optical tomography fits in within the rest of the optical microscopy techniques. I'm aware that, or I'm sure that most of you have used microscopes and are aware of some of these things that I'm going to be mentioning. Here on the left side, you probably are seeing my mouse right now. You have a little dot. It was the smallest dot that I could draw on PowerPoint, which sort of represents the size of a cell. Drawn to scale, and then this is a magnification. These are the kinds of magnifications you get with typical optical microscopy lenses on a conventional microscope, wide field microscope. You can also work on histological sections and get another understanding on how cells organize. You can step up one order of magnitude and then step into what confocal microscopes and two-photon microscopes nowadays can easily image, which is several layers of cells. And what we're seeing here in the back is an animation that shows a reconstruction of a little portion of a chick embryo, which we did a few years ago. So this would be a whole chicken embryo with about 3 millimeters. On the confocal microscope at high resolution, we can get detail about individual cells and their extracellular matrix. These are actually organs, embryonic organs, which contain cells inside. You can get details and reconstruct individual cells. So now you get really valuable information about how cells are organized in vivo and in situ, inside their natural environment, which is something you don't get when you image cells that are on tissue cell culture, like the example that I've shown you previously. And then we step a little bit more into another order of magnitude, into the light sheet realm, where you can actually image whole zebrafish embryos. Zebrafish are very transparent embryos that develop easily in the microscope and show us within 24 hours the full development of an organism. And light sheet is just appropriate for that. It shows us in great detail what goes on inside the embryo. Now, for comparison terms, you have here a three-dimensional reconstruction of a mouse embryo. And as you can see, it's quite larger than a zebrafish embryo. And stepping a little bit more now in another order of magnitude, you have in the back here The quail embryo that actually won that Nikon award that Luciano mentioned before, we're now talking about a few centimeters, 2-3 centimeters, maybe even more. I'll show you examples of that later. So it's just not possible to put these things in a microscope, not even on a confocal microscope, and expect to get any detail other than what you would see on the surface, right on the surface, just a few cell layers. So if you want to understand the full anatomy of these organisms, you need some other technique. And there have been a few techniques, and there are a few techniques out there which can provide you some of this information. One is small animal imagers, but they don't really give you three-dimensional information. They would give you something like a rough image of where a tumor is inside a mouse if you use luminescence, for example. There's another interesting technique. It's known by many different names. One is endoscopic tomography. The idea behind this is to basically cut slices sequentially through a sample and, cut slices through a sample, and as you're cutting slices, you acquire images automatically. And then in the end, you have a full stack. The obvious problem with this technique is that you lose the sample as you go through it, and there are still other problems that can lead to distortions. There are also micro-CT scanners out there. They're based on X-ray imaging. So for us biologists, they have a limitation because we can't really use our stainings and contrasts that we typically use on optical microscopy. And they're also quite expensive as well, as you can see here from the range of prices. Micro-MRI is another example, but we're talking in other orders of magnitude of price. For example, in Portugal, we only have one of these equipments, and it just started operating last year. These are quite expensive. They can give you quite a lot of detail, but as I've discovered, in fact, with optical tomography, you can get detail that even surpasses detail you get with these machines with far less expenditure, I may say, or a synchrotron facility. That's another higher order of magnitude, which is not really easily accessible for biologists. There was, however, an interesting alternative for developmental biologists, which was the Bioptonics 2001 scanner, which was actually sort of a derivation of the original prototype that James Sharp did. And it was being commercialized up until 2 or 3 years ago. And I believe I mentioned that. I'm not sure everybody heard it, but it's just simply not possible to acquire these equipments anymore. You have to either collaborate with someone else or build your own, and that's what I decided to do. So I first have to understand how it works, and I'm going to try to show you relatively quickly what is the principle behind the technique. It's based on the medical CT scanner, which basically puts a human body inside a camera system that has a source of light rays and a camera on the other side. The X-rays go through biological materials very easily. In other words, most biological materials, except probably bone, are very transparent to X-rays, and that's why you see through living organisms very easily with X-rays. Now, that's really not true of visible light, the light we see with our eyes, where not that we are very opaque, it's just that there's a lot of scattering that goes on as light traverses biological materials, and you basically lose this ability to see inside in most biological tissues. Now, through a technique which is a mathematical technique, basically, it's called back projection reconstruction, reconstruct a virtual slice of what the organism looks like inside, and slice by slice. If you move the specimen, in this case the human body, through this beam of light and collect several of these points along the axis of the body, you get a full tomogram, a series of images, like a loaf of bread cut into different slices. Now the visible light version, which James Sharp introduced in 2002, was based on the same principle, but we can actually take advantage of one thing, that we can use cameras. Instead of imaging line by line, we can actually image a full mouse embryo, as you see in this case here, and then do the same process of back projection reconstruction, where each one of these lines, you see here, each one of the lines along the video of the embryo rotating becomes a section, a virtual section through the embryo. It's exactly the same principle. The trick here is to do one thing that is difficult, is make visible light go through the biological material without scattering. In other words, we have to make the tissues transparent to visible light. And that requires a process which is known as tissue clearing. It was developed more than 100 years ago. In fact, I think it was 100 years ago, the first time that a medical doctor tried to see a whole piece of muscle and used methyl salicylate, a very commonly used oil to embed biological materials in. And when you do use oil, you dehydrate the sample, you replace the water by oil, by embedding it in an oil, you get an homogeneous refractive index across the whole sample, which makes light go through it without much scattering. You can still see the details of the organism. It doesn't disappear literally. It's just not scattering light as badly as it was before when it was alive. And now you can actually see through it, just like you see on this mouse embryo. You can now see internal details. So the trick basically is to have a specimen suspended in space, a camera taking pictures of it, a motor to rotate it, generate a movie like this, do the mathematical process of back projection reconstruction, and you get a series of sections. And then you get to the three-dimensional reconstruction. So it really is relatively simple, especially because the mathematics to do this have also been around for almost 100 years. So it really is, as I was saying. What you need is just to learn a little bit of robotics and electronics and have an understanding on how cameras and lenses operate. And being a biologist, I already have pretty much mastered these. Through the help of colleagues, I learned a little bit about robotics and put it all together, and this is what system that I call OPenT kind of looks like, or looked like on the original configuration. It pretty much still looks like this. So quickly to describe what you're looking at here, this is a motor, a stepper motor, and this stepper motor is connected to a magnet which can hold the specimen suspended inside this oil bath. Now this oil bath actually has inside a mixture of benzyl alcohol and benzyl benzoate, known typically as BAB, B-A-B-B. BABB or BAB has been around for quite a while. It was used in the early 90s, I believe, to clear embryos, frog embryos. Frog embryos are fairly large, so they scatter light, and it's not easy to see them on a conventional microscope. If you embed them in BAB, you will see much better through it, and you see several layers of cells. So that's typically what's used on OPT. Samples that are cleared, they are submerged in this medium with BABB, and they're quite transparent. I'll show you an image of that in a moment. You have this apparatus here, which is basically a holder, and this holder has the ability to move the motor, the whole motor and axis, closer to the lens. For this kind of microscopy, and this was something that was confusing for the Nikon jury, was in fact very often with optical tomography, you have to demagnify because samples are commonly larger than the size of the chip on the camera. And the camera sits behind here. For this case, I've used a very cheap camera, €600 camera. It was all the budget that I have. Obviously, if you get a better camera, you get not just better images, higher quality images, but you also it's easier to control the camera and to run the whole setup. The other thing you have here is this. This is basically a LED, an LED light, blue light, that shines through the sample and generates fluorescence. So if on a conventional X-ray system you have light shining through the sample and casting a shadow of the internal organs inside and being captured here by the camera, in this case we can actually operate this system using fluorescence, which is very dear to us biologists. You can just collect the fluorescence here in between the two lens system. You can actually place a filter for fluorescence, and you get just the green out of fluorescence. And practically all of the images that I'm going to show now are autofluorescence images. It's just the fluorescence that the tissues have after you prepare them. So, not a programmer, not really a problem. Micromanager is a software built on ImageJ that is basically fully open source, and it's a multi-purpose software that allows you to acquire and automate microscopes. It's been around for a while. It's constantly being developed. It has a large community where you can find support too. ImageJ, as I'm sure all of you know, is an open source image analysis and processing software that most of us use on a daily basis. And what was really missing from the original publication from James Sharp and his team who developed the original OPenT was to make it fully open. But again, the mathematics behind back projection reconstruction are quite old. If you just Google for something like radon transformation and filter back projection, you get more than 1 million entries, so it's really not a secret. And in fact, there's even one plugin that works quite nicely with ImageJ called radon transformation. It's not perfect. It's not as easy to use as another free solution that's out there, which is very well known among people who already have OPTs. It's called mRecon. It's not open source software, but it's free. It's provided freely. You can go to the website of Skyscan and you can download it and use it to reconstruct your images. So the idea here was that you had to spin the samples, and that required a little bit of automation, connecting a motor physically and electronically to a camera, and building up the system. And it turns out that spinning samples is actually something that's very, very useful. That has been one of the greatest limitations of optical microscopes in the last century, the fact that we're always looking on samples from one point of view. Light sheet microscopy, even light sheet microscopy, can really take advantage of rotating samples and seeing from multiple angles. So why not make that possibility, the possibility of spinning samples, more easily accessible to people? Most microscopy vendors, if not all of them, have ignored this possibility, have not built the hardware or the software to provide this functionality in their systems. So together with a group of colleagues from the Gulbenkian Institute where I work now, we decided that it would be interesting to make this fully open source and accessible to the developmental biologists and cell biology community. So the OPenT, the optical projection tomography open source system that I've built was basically possible, is one of the many possible solutions for an imaging system offered by what we call the open spin microscopy. Now obviously open spin microscopy is a little bit of a joke. Many of you know that light sheet microscopy also exists as an open source project. This was developed by the team of Pavel Tomanchak, and they called it Open Spin. We were working on the same project at the same time, and we contacted them. We knew that they were already working on it even before us, and they were very cordial, so we decided we were going to make this public at the same time. And I'll show you in a moment the publication. And I just want to mention that we already have all the information online if you want to Google for it. Find the information on how to build your own open spin microscope system. The hardware, the software, the instructions on the parts and on where to get the parts, even how to build it. And the website is right here. Or if you just Google for open spin, you will find it. This is an example of one of the movies that you can find there that shows all the parts for the OPenT and from multiple angles and what they look like, how they fall in place. You may find that you want to make a different combination because your samples, and you can even, for example, use a very simple bulb for transmitted light. If you're using large samples, I kid you not, this is what I've used for the largest samples. It seemed that getting the unidirectional source of light was not the ideal. You create lots of shadows. With a very diffuse light, you actually turns out you get better results with OPenT. It's been quite an interesting project, getting to know the system, perfecting it to get the images that I'm going to show you. So this is the small correspondence that was featured in Nature methods last year, which the reference to it is here. This is the example of the OpenSpin system, as you know, as I mentioned. Pavel Kamenchak also has developed this, and even Corina Lorenzo has done in his online versions of how you do your own spin system. So one of the advantages of the OpenSpin system, the platform that we developed, is that it's really fully open source. All of it is fully open source. And you can get even other combinations of it. And I'm not going to tell you more details because Emilio, the person who developed the plugin itself, will give you the full details of it in a webinar on, I'm not exactly sure whether it's the 16th or the 17th. I believe it's the 17th, but the Bitplane guys will make sure you know the right date and times. So how do you operate the OPenT system? And there's actually two simple ways of doing it. One is by just using the sketch. This is built on Arduino, and for those of you who are not familiar with Arduino, Arduino is basically a microprocessor board that allows you to control, very easily control and make software to control things like small robots and motors and lights, which is just perfectly suited for this project. It's very simple to program, and little programs for Arduino are known as sketch. You need to get a camera that has a trigger that accepts an input trigger. So the Arduino basically tells the camera to acquire an image, tells the motor to rotate another angle, acquire another image, and that's just what you need to automate while you're acquiring images. And this is important because if like me, some of you get on this project to use an old camera, or you only have money to get a cheap camera, the cheap cameras very often are not recognized by Micromanager, so you're stuck to this solution. But if you're lucky to have a really good camera that's fully controllable from Micromanager, you can actually use the Micromanager OpenSpin plugin that we've developed. And there's an option for it here for OPT operation. You basically state here what is the rotation, the number of rotation angles that you want. Acquire. The other advantage of the Micromanager OpenSpin plugin is that you can actually build a system with multiple light sources and get multiple channels. But if you're just using the simpler version that I'm presenting today, you can just have this Arduino sketch. And you're very welcome to send me an email. I'll send you the sketch. In fact, I believe it's online on our website as well. It's a relatively, like I said, simple thing. It just automates the process of acquiring the image, rotating the motor, acquiring image, rotating the motor, and it just runs on a loop and on a continuous loop. I leave it running. I acquire at least 1,600 images, which corresponds to a full 360-degree rotation with my stepper motor. And after that, we move on to the filter back projection reconstruction and get the 3D data set, as we call it, which is basically a tomogram. So how good are the images after you've reconstructed the tomogram? Well, here are some examples. This is a reconstruction of an E14, and you seem to have lost the video now, not the sound, but the video. Okay, we're back on. So this is a three-dimensional reconstruction of a mouse embryo, E14, 14 and a half days mouse embryo. And as you see here from the sequence of sagittal sections, you can see all of the internal details, all of the anatomy, internal anatomy details of this 6-millimeter sample. You can actually reconstruct and see here using volume rendering, sophisticated techniques for volume rendering. You can actually see internal details, and you'll see an example of that later on Dieter's presentation. When you're acquiring, with my 2-megapixel camera, you acquire a data set which is about 6 gigabytes. You need quite a lot of, you don't need a very powerful computer, but you need a lot of RAM and hard disk space. After you've done reconstructing the tomogram, you end up with a data set that's typically 1 to 2 gigabytes. So it's still fairly large. You can reduce it and still get a lot of detail from what's going on inside. Another example is this. This is a data set from Seth Ruffins from the Caltech, which they've acquired using micro-MRI, and they had a project to build a database for quail, several stages of development of quail. So this is the kind of quality you get from a micro-MRI setup that costs millions of euros. However, using an optical projection tomography like the one that I'm talking about, you can get fairly similar images with detail that really practically rivals what you get with a micro MRI, and this has cost me less than 3,000 euros to build. In fact, the images are so good that the Nikon judges liked it and decided to award me the first prize on the Nikon Small World Video Contest Store in motion in its contest last year, which was a great pleasure to me and a great honor. So this was the winning video. It's basically a sequence of slices. The embryo, the 10-day quail embryo is assembled slice by slice. It's about 1,000 slices that you acquire, and it's volume rendered one section at a time, basically, and builds up. One more advantage, the fact that you build your own system means you can actually customize it. Now, the original optical tomography scanner that's no longer being sold was prepared for samples that are relatively small mouse embryos. Rob, Bryce, and Richardson from Monash University in Australia was working on a full atlas for quail development from the early stages to the later stages. And he actually had access to an OPenT 2001 scanner. And he could only image embryos on that scanner up to about halfway through, or not even halfway through full development. After this stage, which is about halfway through development, the embryos are just too large for the commercial OPenT. And Rob knew, and we've collaborated on imaging courses in the past, and Rob knew that I was building this system, and in fact he gave me some advice on how to optimize it and build it. And he knew that I had this huge chamber because I wanted to have something that would accommodate all types of samples. And he sent me really large embryos. Like this embryo that you're seeing here, you see the green fluorescence as blue light shines through it. It's all embedded in DABB, the clearing agent. And notice that you can actually see through it. It's transparent. It's almost like an X-ray through it, which you can do with visible light after you've cleared the embryo. The embryo is inside an agarose block. You can see it. It scatters blue light here, so you can actually see a little bit of the agarose, but once the camera captures it, and it's filtered out by the emission filter, you don't see it anymore, and then it's glued to a little piece of glass, which in turn is glued to a piece of metal. And this metal washer connects to the magnet and the motor. So in fact, when this is operating, the embryo is flipped 180 degrees and it's suspended in space as the camera is acquiring. And what you're seeing here is something equivalent to what the camera would see when it looks on the sample. So again, this is that quail embryo that won the Nikon contest, and that is about 2.3 centimeters long, fairly large. And just for comparison, the mouse embryo or a zebrafish embryo. Rob really wanted to get as far as possible, so he sent me embryos, quail embryos with 13 days and with 15 days. This is just one day before the quail embryo is born. And I couldn't believe it, but it's actually possible to get some decent images of a thing that are as large as this. This embryo is about 4.1 centimeters long, if I believe. This is the largest I've ever done. It's 4.3, I think, centimeters long. It's a mouse, it's a rabbit embryo near to term. It was about to be born as well. And you still get quite a lot of detail on the internal anatomy of this beast, this huge microscopic beast, to call it that way. You can also use bone stains like alizarin red and alcin green. This is what the camera sees. You get 360 degrees and then you get a reconstruction, and you can see from any angle. You can actually quantify, isolate individual bones or cartilages and see. So this is a project that one of the PhD students here and at the Gulbenkian Institute is working on, and he was very interested in this technique. What he had to do was actually resort to a synchrotron facility, and he no longer needs to do that. He can actually do it here on the OPenT. Another example is, for example, it's a paw of a chicken embryo, which you can acquire. This is what the camera sees using transmitted light. So it looks exactly like an X-ray image if you use transmitted light instead of fluorescence. And then after reconstructing the images, you get these slices, which you reconstruct in 3D. You're still looking at a sample rotating in space, but now this is actually a three-dimensional data set. You can actually measure volumes and distances with high accuracy. Or this is an example of a flower that came from Brazil from a collaborator at the University of Lisbon, where I started this project. It's a 2-millimeter flower that they wanted to see the internal details of. So you can get some really interesting images with this system. So what do you do with these data sets besides just generating movies of things rotating in space? Well, you can inspect them in 3D section by section. You can do 3D renderings, which are basically what I've been showing you. It's a virtual reconstruction of what the object looked like if we could see, if we were diminished to the size of that object and we're looking directly at it. And there's a lot of freeware software out there to do that if you don't have commercial software, or you can use, obviously, MRs to do that as well if you already have access to it. But there's other things you can do that the software allows you, is to get very accurate morphometrics of morphogenesis and morphogenetic processes. How large is an organ, specific organ, for example, the heart, or a specific blood vessel, throughout development, how it changes throughout development. You can do it in a very quantitative manner using this technique. And you don't have to just really destroy the samples or physically alter them to do that. The other big advantage is once you've acquired a 3D data set, it's there. You can go back to it, go back to the computer. You don't have to do other experiments unless you actually need to do several experiments to get a significant N. But the great advantage of this is that if you really just need a three-dimensional atlas, once you've done it, you already have it. So we generate these really beautiful data sets with a lot of internal detail of the anatomy of these embryos or whatever other specimens you're interested in. It could be, for example, the brain or the heart of a mouse that you're trying to look at and compare with a mutant. So what do you do with these data sets? It's a shame to just use one or two images, put it on a paper, and that's it. You can actually share them. And there have been many examples, very good examples of projects where people basically created the conditions to share digital atlases online, digital information or 2D data sets. But probably the most, the longest and best known example is the Mouse Atlas project that originated in the UK, in fact in the place where OPenT was developed. There are other ones like the Anatomy Portal, which already has the zebrafish and the quail anatomical data sets online, OpenWarm, Digital Morph, many others. The level of openness varies. And sometimes the easiness to which you can get to the data varies quite a lot as well. Some are so comprehensive that it's not very easy to get just to the images and just to get the data set itself, to learn to do two-dimensional reconstructions or just get cool images. And that's where Haeckaliens comes in. Haeckaliens was the way that I figured out with little resources to share the data sets that I was working on. It's actually a web page that I started. And let me just tell you a little bit of why I got into this and why I'm so interested in anatomy of late-stage embryos. Most of you, if you are a biologist, I'm sure have come across this image, which was an illustration on Ernst Haeckel's books in the late 19th century. This picture fascinates most of us. In fact, it fascinated me, and it was one of the reasons why I got interested in embryology and evolution. We all know there is a fallacy behind these pictures, and that he tended to accentuate certain characteristics of embryos to support his ontogenetic recapitulation theory. And I mean, we're all aware that there were flaws in his interpretation, but the principle and the fascination that his images have is still alive today. And one of the things that once I had access to optical projection tomography, one of the dreams that I wanted was to fill this plate with actual images of embryos and try to look at internal anatomy, because most of what we have done of comparative anatomy of embryos has been just from looking at external anatomy. Now we can actually have access to internal anatomy. So Haeckaliens is a website, and the website is here where you can get these data sets that I'm working on. You can play with it yourself with your software. You can download them, or you can get examples on how you can share, if you do images yourself, if you have one of these systems, on how you can easily share three-dimensional information as supplementary materials for a paper. Because sharing three-dimensional information on a scientific article is not very simple nowadays. It may be simple a few years down the road, but it's still not very simple. So the website is actually featured so far by Google Sites, and it's hosted in my personal webpage. It contains 3D renderings like the ones I've been showing you, data sets that you can download and use yourself, 3D animations of things that I've done that I found cool and interesting, and I just put there to inspire other people to invest in learning 3D imaging and reconstruction. An example of an interactive 3D viewer where you can actually see in 3D inside a web page, or even Imari's versions. I've started putting some of the data sets, so if you're into Imari's, you can actually download some of the data sets. Or 3D PDFs. I don't know if you're aware, but you can actually create PDF figures that are 3D, that are interactive. You can actually click on the image and rotate and show the internal anatomy. Before you do that, you need to actually segment and reconstruct organs, which is something Dieter is going to talk about in a moment, how you can do that in Imaris. So I also want to say that I'm open for contributions. If you have embryos and you don't have this technique, I'm really willing to collaborate. You can send me the samples or come visit us and see the equipment. Or if you want to actually, if you already have the data sets and would like to contribute, you're very welcome. These are the size and the types of data sets that I'll be welcome to put on the website, very happy to put on the website. Just keep in mind that what I'm trying to do now is just get mostly non-model species, because there are already excellent sites out there for model species. So I'm mostly interested in non-model species. And just to give you an example, so this is a rabbit. You just click and you get access to these types of images that I've shown you. And I also every now and then provide some cool reconstructions just to really inspire people, just get the wow effect basically, which, truth be said, it does make a difference on a publication. It does make, having that wow effect on your images does really help other people draw other people's attention to your work. You can also use YouTube to publish movies and animations in 3D. I don't know if you're aware of it, but there's that option. You have to learn a little bit of how that's done. Some softwares like Drishti can actually do side-by-side stereo pairs. At this moment, I'm not exactly sure whether Imaris does it or not. I believe it does it as well. So if you do a side-by-side stereo pair movie, just like the ones that are broadcast on 3D TVs, you can actually broadcast it as well on YouTube. And if people have 3D glasses at home or are lucky to have a 3D monitor, they can actually see it in 3D. Or, like I told you, the WebGL interface to put a 3D image on a website. You can do this through a website known as SliceDrop. You can actually go see slicedrop.org and see how this is done. And again, a 3D PDF, this is what a 3D PDF image looks like. And you can actually have, if you've segmented the different organs, you can actually turn them on and off and just see parts of the embryo. This is a reconstruction of that rabbit embryo. So just to give you ideas of what you can do with these data sets, don't just have them in your lab, share them openly, because you may not just be saving other people a lot of hard work by trying to recreate things that you've done already, but it's actually a good manual for anatomy that students, even high school students interested in anatomy and embryology, can actually take advantage of it. So that's one of the things I like about the open source aspect of it. And I believe now I would move into Dieter, invite Dieter to show us how you can get these amazing images using Imaris also. I just would like to finish by thanking the people who I've collaborated in the past. Obviously, Bitplane for hosting this seminar, and all of you for your attention, and all of the people that I'm collaborating with and that I've been collaborating with. A lot of them are not here yet because I haven't even shown you examples of what we have done, and also some of the funding agencies who have paid my salary in the last few years. And obviously the Gulbenkian Institute and the Advanced Imaging Unit, which I'm now running, which are hosting this project as well, which I've started when I was at the University of Lisbon, and now I've moved in here to the Gulbenkian Institute.
So now we have Dieter, and he's going to show you a couple of slides, I think only really a couple of slides, and then move to Imaris itself and spend some time there to show some of Gabby's images being segmented and analyzed and how they can be fully analyzed using Imaris.
Yeah, hello from Germany. This is Dieter. So I don't plan to show a lot of slides as just only an opener. Let you see my name and as well my email address. If you have more questions on how to use Imaris on that data sets, you can write to eusupport@bitplane.com. Okay, so with this, I just want to go right into the software. So the output, or where we would take over the data set, is when all this reconstruction was done, and you have a stack of slides, orthogonal slides, normally in a TIFF format, and this you can read into MRS. And this we see here, just the TIFF slides loaded into MRS. So this is a simple slider view, so I slowly go through the slides. You see this is the bottom outside of the embryo, and then we go up all the way through the layers. And in this case, we have here 480 layers in the data set, and we have an X and Y amount of pixels of 777 by 600. So that's about a 200 megabyte data set which we are looking at. Okay, the slides are nice, but and beautiful already by themselves, but of course we want to do more with this. So there, I want to show you some other 2D visualization tools which we have inside MRS. So that would be, for instance, here the section view. Probably most of you know these kind of visualizations. So we have here the original cuttings, the layers, and then you can move up and down through the layers with these horizontal line. You see that this updates here the X Z view. So this is the X Z view, this is the X Y view, and this is the Y Z view. So you can travel through this and have a first inspection of the data set. So from here, of course, we want to go further into real 3D because this is what MRS is built for. And so we move to the next visualization tool, which is called Surpass. And inside Surpass, you get now already a 3D reconstruction by default. So if you open this, all the layers which we have seen before are brought together, and you get a 3D reconstruction. So what I do now here is always live. It's not pre-recorded or so. Always. So here you can rotate this, for instance, and look from different angles. As well, you can do very relative simple things like changing here the contrast, so you can remove darker structures from the image with this triangle here, which sets the minimum value which is displayed. You can ramp up the contrast if you want to have this brighter. And with this gamma correction, you can ramp up the dark intensities from the data set. So it gets just nicer to see. Okay, so there are several ways how you can do this volume reconstruction. One way is you can use here the blend mode, and another mode would be, that's what we see now. Here we see mainly the outside of the data set. You can play a little bit with transparency so that you see as well inside. This is a transparency slider here. But the main purpose is that you see opaque objects and here the outside of the embryo. Another way to show here would be the maximum intensity projection. This is just a different visualization mode which allows you to look inside the object, inside the specimen, and here we see just the bright intensities along the way. So it's not all the dark ones which are outside, but mainly the bright ones, and so you can easily see what's going on there. Okay. So from here, the next step would be to get now more information about the different objects inside this. So, for instance, if the question is what is the size of the heart, what is the size? Sorry, there was some ringing in the background. That was on my side, so sorry for that. If you want to have these objects separated from each other. So in the moment for the software, this is all just one cloud of voxels, and you don't get really statistics on this information. For this, we mainly use a tool which is called surfaces, and with that one, you can build segment individual objects. So I have to admit, I did these segmentations. I'm not an anatomist, so I'm coming from more from the genetics and cell biology area. So bear with me if there are some errors in the segmentation. But it should just show you what you can get. So what we see here are the different organs which I have selected to segment. We have here on the list all these objects as well available, so you can click on them, switch them on and off, and look at objects which were maybe before hidden. So for instance here, this one. You can as well play around with these objects by changing, of course, their color and their transparency. For instance, if I select here this outer brain part, I can change here the transparency. So if you have inside objects, inside the outer ones, here is just a more or less random segmentation which I did on brightness. So you see just the bright parts inside this forebrain here shining through, so you can combine this. And of course, as well, you can change simply the color of everything which you want to highlight, maybe. Okay, from here, the next step would be, for instance, to ask now the question: okay, what is the size of this part? What is the size of the heart, for instance? And for this, you can basically just click on the heart and go to the statistics, and in the statistics area you will be able to look up several statistics. For instance, this is here now the surface area of the heart, which gives you the complete surface area, or other simple statistic would be the volume of this heart. And there are more statistics which are not all calculated in the moment. There would be as well the size of the smallest box which you can put around it to get automatic information about length and things like this. So these kind of statistics you can get easily from the segmented object. Another statistic value which could be of interest is, for instance, the distance between objects or between certain positions on the object. So this is for this we have a tool which is called measurement points. And measurement points works in the way that you can manually place anywhere on the surface of the object, or as well earlier on the volume, if you like, but it's on the surface. It's normally nicer to use. So I position my mouse pointer here, and this little cube you see will jump on the position in 3D space where you want to start to measure, and you press Shift and left mouse button, and on the other side as well, and you will see you get a line which shows you this distance. And of course, all these statistics you can then later export here with this little floppy to files, Excel files or CSV files, for instance. Okay, another thing which is quite cool with Amaris,